Good morning to all and welcome to the MNS monthly meeting for February 2022. Request everyone to please accepting, Sudhakar and Geetha and Dr. Subramanian and Kumaran to kindly switch off your video and to mute your mic. As Sudhakar was saying, the Asian waterbird census is on full swing. Started last week. As usual, we 6 teams fanning out across Chennai and its hinterlands. So even today, there's a team. There are teams, and I do find quite a few of them are also, picked in. So probably listening into what is going to happen. Listen to Dr. Subramanian, and it's a very also on the 27th of this month, like last year, we are having the 15th edition of the HSBC Chennai Bird Race. So quite a few teams have already registered, and we are hoping to have more teams registering. We are restricting the number to about 20 teams, and it's also restricted only to MNS members, their families, and friends. So, with this very brief update on what is happening, this is a very, let's say, a very, very important event that is going to follow this morning, and to start off the process, I request our President Sudhakar to deliver his opening remarks.
Good morning, everybody. A warm welcome to you all. This is a day of pride for MNS in the sense that we are going to bring out a special issue of Blackbuck, which is called the Moths of Tamil Nadu. So, first of all, I am very thankful to Dr. Subramanian to agree to release this. Who is the most suited person to release this, because he has been involved in moth, and he is himself an expert on moth, and he is involved with, he is from ZSI, who has undertaken a lot of moth expeditions. I'm also thankful to Dr. Geetha Iyer, having worked so hard to provide actually about 350 species of moths as photos, and about 1,319, I think, the list of moths which have been recorded from Tamil Nadu. All this is a very tremendous hard work. The experts will probably talk about this, and they will know more than I do about these things. A Blackbuck, which has been in existence from 1985 onwards, you will see this is volume 37. So this is in the 37th year of publication. MNS has always like to put more people, get more people interested in wildlife and take interest in their surroundings, and thereby promote conservation. It is best that I actually quote Geetha Iyer, who says in an introduction, this will be the most suitable introduction for this publication. She says this is written mostly for lay people, which we are, to introduce them and help them with moth watching. So the intent here has been so that it will be accessible and understandable to the wider community, from a class 7 student to a university student, and adults who may not have a science background, but yet are enchanted by moths. So with this brief introduction, I think this is a very apt quote from Dr. Geetha Iyer to begin this function. Thank you.
Maimoon, I request you to kindly Mute your mic, Maimun. Yeah.
They changed the timing.
Okay. Now, Acha, we have to go to. I think the host can mute most people if somebody is not muted.
Okay. I now request Kumaran to introduce our chief guest, Dr. K. Subramanian. Kumaran.
Good morning to everyone. It's my privilege to introduce our distinguished guest this morning. Dr. K. Subramanian is a scientist with the Zoological Survey of India, which is also referred to as the ZSI. And he is the officer in charge of the Southern Regional Center of the ZSI, which is located at Chennai. Dr. Subramanian specializes in the taxonomy, ecology, and biogeography of aquatic insects. Specifically, he specializes in the Odonata, Ephemeroptera, and aquatic Hemiptera. He also specializes in biodiversity databases and GIS applications in biodiversity research. Subramanian, Subbu, to those who know him, has published 8 books and over 140 research papers in international and national journals. And he has described many new taxa of Odonata, Ephemeroptera, Hemiptera, and Zocoidea. He is a member of the IUCN Odonata and Invertebrate Species Specialist Group, and he is a recognized research guide at the University of Madras. Currently, he has 6 students pursuing their PhDs in various aspects of invertebrate taxonomy and ecology under his guidance. I would like to add that Subbu, Dr. Subramanian, started watching birds when he was just 5 years old. He started watching birds at Palakkad, his hometown, and today his cumulative bird list from his home numbers more than 120 species. So he's a naturalist, a passionate naturalist, even before he became a scientist. And one of the major contributions, I think, that has touched me definitely is that among his books is a pioneering photographic guide to the dragonflies of India. This book has played a significant part in triggering widespread interest in dragonflies of India everywhere in the country, and I suspect even in countries around us. Dr. Subramanian routinely travels to locations all across the country in the course of his work, and he conducts field surveys, faunal surveys in these locations. He has a wealth of knowledge and experience. He shares it very freely with other naturalists, and to have a conversation with Dr. Subramanian is to have a spirited discussion on various aspects of natural history. So, ladies and gentlemen, Dr. K. Subramanian.
Thank you, Kumaran. Thank you, MNHS, for inviting me to this function. I'm very delighted. And Kumaran is my very old friend, so I don't know when we met first, but we have been communicating for a long time, but maybe last 15 years, but in various ways. And we have done some fantastic project also. One was that I should mention Kumaran helped us to digitize all the publications of ZSI. Now it is online. And I'm very delighted that my friend Geetha has made this wonderful book. And I also know Geetha for a quite long time. And I've been a regular reader of Geetha's articles in Frontline and other places. And I always remember, like when I see Geetha's article, many of you may be aware of that Jean-Henri Faber. So Jean-Henri Faber was a French school teacher, and after retirement, he took up so he was with his modest savings, he took up a place in northern France, I think, and he started watching insects and started writing about it after retirement. So though he was writing before that, but his serious writing started after retirement. So they are beautiful prose, like you should read some of them. Read, and I think one of the first article I read about by Faber was his article on peacock moths. So it is called The Great Peacock Moth. So it's a beautiful prose, and I think that is he does also experiments with the insects. So they are very good experimental animals. So he's the first person to show that that antenna is very important in insects for detecting pheromones. So he has done beautiful experiments with the great peacock moth. And so whenever I see Geetha's article, I always remember that, okay, Geetha is our answer to Jean-Henri Faber. So she also has a lot of similarities in her career. But this her recent publication on which we are going to launch today is a beautiful compilation on moths of Tamil Nadu. So to my so many of you may be knowing that Geetha have been working on this for several years, and she has compiled it as a beautiful photographic field guide, and this will definitely inspire a lot of people to start watching moths. And before Geetha, so the recent compilation of moths of Tamil Nadu is about 1,100 species. So Geetha has compiled 1,300 species. But if you see that moth studies in Tamil Nadu is not new. So it's from Britishers' time, the Hampson, who wrote the Fauna of British India, 7 volumes of moths, Heterocera it is called, and was working as a tea planter in Nilgiris. So he, in the Nilgiris alone, he documented 900 species of moths. Of that, 300 were new to science. And all these are published. And there is a separate volume on by British Museum. It is called the Illustrations of Heterocera in the British Museum. So there is a volume by Hampson, or I think it is volume 7 or 8, that illustrates the moths collected, color illustrations of all the moths collected from Nilgiris. So that illustrates about 900 species of moths. But of course, the taxonomy is changed. So it is more than 130 years. So the taxonomy is changed and a lot of nomenclatural changes are there. But still, it is a valuable contribution to moths of Tamil Nadu. Other than that, Frederick Moore, who also wrote the Lepidoptera indica, so has done contributions, then Bell, then Sinhu. So these are all the people who pioneered moth studies in southern Western Ghats, especially Nilgiris and Animalize. But interestingly, many of these specimens were also collected by Indians, especially the Nathan family. They were very good insect collectors, and they supplied specimens to all these foreign nationals and foreign museums. That is how we got to know more about the moths of our part. But recently, after the especially after the internet, a lot of people have started watching moths, especially from India. And there were some good studies. Kerala, there are published checklists. I think 2018 or 19, there is a checklist of moths published by Kerala. There is another MNHS in Kerala. It is called Malabar Natural History Society. So they also have a journal called Malabar Progon. So that published an article on special issue on moths, and that has a checklist of moths of Kerala. And I think Subbalakshmi in Northern Western Ghats also published moths of Northern Western Ghats. So these are some of the recent publications. But my association with moths is mostly photography. So I also put light traps to collect many insects. So say many of my friends study moths. So what I do photography for them, and I give them, especially Sanjay Sombi and Naveen sorry, Navaneet and all those people, I view them. So sometimes we go together in the field. So they will be collecting moths, so I'll be photographing for them. So I have a good collection of moths photographs from different parts of India. So most of it is I have given to Sanjay or Navaneet for their publications. And so that is my association with moths. But interestingly, there are some aquatic moths also. There is one Pyralidae family. So that has at least some 80 species of aquatic moths. So the aquatic moths in the sense that the larvae of the moths, they feed on aquatic plants. So some of them are, they live underwater. So I have collected them even in Western Ghats. We have a genus called Alocaudus. So that lives underwater in the streams, and they make tunnels in the aquatic plants, especially the algae and other benthic plants, and they live there. And during summer, you can see them like in the rocks, you can see them as white patches, they pupate and they emerge in large numbers. So there are some interesting moths also. So if you look at moths of Tamil Nadu, currently about 1,300 species are documented by Geetha. But if you look at the plant diversity, so moths are closely associated with plants. So nearly 4,000 plus angiosperms are there. Flowering plants are reported. So even if you a modest estimate of like each species of plant has at least one species of moths. So our associated moths, so we may cross even 4,000 species of moths, and we know very little about their natural history. So most of the things what we get is through light trapping, and what the moths are get attracted by light traps. But the interesting thing is that we don't know what is their food plant or how they live. So very few people are trying to understand all these things. So there is lot of things our Madras Natural History Society members of Madras Natural History Society can do, because they can watch moths. Because most of the moths, can many moth species come to home, or they breed in your garden. So you can see them, especially so they look repulsive. Of course, the caterpillar look repulsive for some species, but their natural history is very, very interesting. So there is a lot of things available online, and there are a lot of people available online to help. So there are good websites. So Geetha has mentioned everything in the book. The beauty of the book is that it's all beautifully illustrated with color photos. And I think she's illustrated 324 species of moths, illustration color photos. And there are about she has given a nice introduction to moths, and there are how one can go about moth watching is also mentioned. The body parts are mentioned. So it's a very good beginner's guide, especially for naturalists to start moth watching. So of course, many of the MNHS members are birds. So an important food for birds are moth caterpillars and adult moths. So it's very, very important to know that what are the moth species we are having. And we can do a lot of natural history studies and life history studies, especially. So we can rear moths at home. So that is the best way to know their food plant and also to know their natural history. So with this brief introduction, I launched the book to all of you. So this is, I think, the book, Butterfly. So this is the book, and this has, like I told you, this has nice illustrations, color illustrations. All the species are in a name, and you have, like, she also talks about polymorphism, and she has illustrated some polymorphism, some interesting facts about moth. So this is a very handy and very nice field guide for moths. And at the end, she also talked about a detailed checklist of all the 1,300 species of moths that she has recorded. So this is lot of things. So we have another, we have to cross 4,000 plus species. So that with all your efforts, I think all your natural history efforts. So most of our studies have been restricted to Western Ghats. Of course, that is rich in biodiversity, but plains of Tamil Nadu and coastal area have a lot of species to describe. So with this, I congratulate Geetha for this wonderful contribution to all of us. Thank you.
Thank you so
Sudhakar,
very much.
Vijay, may I add a word?
Yes, please. Please.
think
Just
we-
before that, Lakshmanan, I think your video and your mics are on. Please mute both. Thank you. Yeah, Geetha.
Okay, thank you. I wanted to thank Dr. Subramanian for his generous words. Especially I'm very touched to think that I could come even anyway somewhat near to what Henry Faber did. The amazing work. I think everybody is quite aware of he's like God of insect study for me, So thank you so much. And as you mentioned, there are a lot more things one can put into a book of moths, especially the moth plant interaction, about which I haven't touched at all. There are a whole lot of things that can be added to this, but given the space constraint, I had to restrict myself. And yes, aquatic moths, the larval studies. I think there is one MNS person, Yuvan, who actually breeds moths. So he's the one person who's done a lot of breeding, and so he has some very interesting life history observations on moths. There are some young people across Tamil Nadu who are very much into detailed studies. So we hope that we can get to know a lot more amongst the Eastern Ghats. People do a lot of work at Western Ghats, but Eastern Ghats diversity is also very interesting, and we hope more can be done. And I wanted to thank everybody. I see a lot of congratulations on the chat box. Every one of you, thank you very much. I hope this will be of some benefit. Thanks.
Thank you, Geetha. I will now take you all through the, through the book. I'll go page by page. I'll just scroll down and just give me a minute to share screen and start. I hope the screen is visible. Yeah.
Yes, the screen is visible.
Yes, yes.
Yeah. Okay. So this is the wrapper, and of course this is the special issue of Blackbuck, volume 37, numbers 1 and 2. So the contents is the moths of Tamil Nadu, as Dr. Subramanian said, 350 moths photographs, close to 1,300 species listed in the checklist. And all the photographs been taken by Geetha. So Geetha talks about, all the background to the whole book in this chapter, and then takes you through the various forms and describes very clearly what the moths are. And then there is then the pictures begin. The soft copy of the Blackbuck issue will be shared on the WhatsApp group and our MNS Google group immediately after this meeting. So you can all leisurely go through, download it and go through. It's a tremendous amount of work that has gone in. And Geetha has also acknowledged the assistance of 2 of our members in helping her with this. And one is Vikas Madhav and the other is Mahati. You're struck by the different, the shapes and the colors and the amazing. I mean, moths have always been a fascinating taxa. I think I'm sure a lot of people will develop fascination for this.
Vijay, can you just stop for a moment in that page?
Yeah.
You see, there is, despite being very careful, recently there was a revision and there was some correction done. And so this particular moth 276 Autocaris phasalis, I want to make a mention that this identification is incorrect. It's actually Autocaris amethystina. I requested Kumaran and other editors to carry an errata for this particular moth.
Okay.
276. I just want to bring it to the attention of everyone.
Sure. I thought they
So much.
changed. I
Huh?
thought I thought they only change the names of birds every year.
No. Moths,
I
there is
know.
so much more discovery
I
that
know.
if you ask Kumaran, he will tell you how much headache I have given him as he edited through copies of it. Every now and then I would say, can you please change this? Can you
Yeah.
please add to the list? I think I drove the editor slightly crazy with all the corrections I had requested to them. Thank you so much, Kumaran and Sudhakar. Okay, you can continue.
Yeah. So this is the checklist. It's about 1,300 and odd. And you'll find this name Hansen and Clark and Warren regularly being quoted. Yeah. So that's it. This is the back cover. So this is a new and undescribed moth from Kalakkad Mundanturai Tiger Reserve. Well, this is fantastic. So let's stop sharing. So thank you all very much. And if there is Dr. Subramanian, do you have a bit of time to take questions if people have any?
Yeah, sure. I think Geetha will. Yeah. Can
No.
moderate.
No, no.
Yeah, I mean, if there's something I can add, I'll definitely add.
Yeah, just that if there are any of the participants who wish to specifically, and incidentally, there is recently Dr. Subramanian published a book on the aquatic insects. There's tremendous. It's not recent. I think it was, but it's a much used and much referred to book. He was mentioning about aquatic moths. So there is a fantastic book written by Dr. Subramanian on aquatic insects. I think if you were interested, should be able to, I'm sure, find a copy at ZSI office. Yeah.
No, it's only available as PDF copy, never printed.
Okay, all right. But it can be downloaded from the.
Yes. Yeah. I think that is also outdated now.
Oh, is it? Okay.
Yeah. It's published about 10 years ago. It's outdated now.
Oh my. Okay, I know. The oceans are really churning. Right?
Yeah.
And so are our waters, which retain wherever they are supposed to be. Anyway, so members, if any of you have any queries to ask, you could put them in the chat box and I will go through them. Of course, there's a lot of congratulatory messages for Geetha.
Yes, which I thanked everybody for it. Thank you so much.
Yeah.
Can I, Dr.
Yes.
Can I ask a question? No, he was talking about aquatic moths. I was just wondering, Dr. Subramanian, whether these are specific to certain streams or Western Ghats or Eastern Ghats as aquatic insects. These species, are they endemic to certain areas? That kind of things are there for the aquatic moths, or they are probably found all over India. These aquatic moths.
No. See, there are these spiralid moths. There are several species that use aquatic plants. Okay. So, and the stream ones are like we get in Western Ghats. Most of them are restricted to streams. But there are other wetlands also. There are aquatic moths, which are like which can feed on aquatic plants. But those which are found in the other kind of wetland, like for example ponds and lakes and other things, they are widespread. So some of them are pest also. Some of them are pest of the paddy and other species, but
especially paddy, because there's so much of wetland associated with it.
So some of them are pest of paddy, but the other ones which are found in streams are mostly restricted to hill streams. But they are also not endemic. They are all widespread species because they have
widespread also. Perfect. So this gives us one more reason to, constructing check dams and diverting Western Ghats streams and all that will have an effect on these aquatic moths. So this will be probably affected by all these things. Gives us one more reason to protect these water streams and water bodies, I think.
Yeah, but see, like if you like to convince the policy makers, these moths and insects and all, we won't sell.
So that's a tall order. I think so.
In fact, DST has reduced money funding for taxonomy work, isn't it? Am I correct, Mr.
Yeah,
Subramanian?
yeah. See the like the
Department of Science and Technology won't fund taxonomy work, especially
So
invertebrates.
it's not like the stream ecology. If you see, it is not aquatic. There is a whole fauna and flora associated with the riparian system. So it's entire ecosystem. So, and there is a lot of studies. So it's not just that there are animals only in water. There are other things along the streams which are called riparian zone. So a lot of amphibians, reptiles, even birds. So unfortunately, like we don't have many typical stream dwelling birds. So if you go to Himalaya, there are a whole lot of stream dwelling bird communities there. So Western Ghats streams like that way, they are very poor in bird. On other than you can say whistling thrush or some wagtails. But if you go to Himalaya, you have forktails, you have dippers, you have all these red starts and all those things. So they are entirely dependent on stream and stream insects. So that kind of bird communities are not there in Western Ghats streams. So though they are evolutionarily much ancient than the Himalayan streams. So that kind of the bird associated with streams are not.
So,
But in Himalayas, that kind of streams.
Mr. Subramanian, I have a question for you.
Yes.
This is regarding, I wanted to know, I'm on several mudflats in Mumbai, and they say that there is an insect or algal population or crustacean population that the flamingo thrives on. Could you highlight on what exactly is it feed? I'm inquisitive to know what is the major component of its feed.
Okay, so you know that flamingos are filter feeders, like they feed on zooplanktons. Okay, so it is not. So it's a soup actually. It's a soup of zooplankton. So zooplankton is not a plankton itself is not a taxonomic group. It is a anything that any animal that can float and move are plankton. So you have larval stages of several groups. So even you have like including like crustaceans, mollusks. All those things have plankton form. So the flamingos feed on that. So they have this. They unlike other very interesting aspect of flamingo is that. So usually in birds the upper beak is longer than the lower beak. So if you look at the bird morphology, but in flamingo it is reverse. So the lower beak is larger than the upper beak, and that is why it turns the head down. It turns the head down and it feeds. And it just feeds by filter feeding. So the flamingo beak has combs and through which it takes all these zooplanktons. And the color of flamingo comes from this zooplankton. So you have this what do you call that? Which produces the color. So like
Ketognath, ketognath or any other form of zooplankton.
No, all zooplankton. It feeds on all groups of zooplankton. So,
Carotenes?
Carotene, beta carotenoids. So it has a lot of beta carotenoids. So that is how it gets the color. So even many birds get the color from that. Even our hornbills, great hornbill, get the color from this.
We don't have insects. What about insects?
So actually, like if you look at it, there are not many marine insects. There are purely marine insects, there is only one, so that which live in pelagic area. So that's a strider. So it's called halobates. So other than that, you have coastal insects. So but or estuaries, but purely marine, there are no insects.
I have a question for
Thank you.
either of you. Yeah.
In fact, it is said that because there are no insects in the oceans, they are replaced by a lot of these crustaceans and other jellyfishes and those kind of slugs and all that. Basically, a lot of crustaceans that are present in the ocean has some evolutionary significance in the absence of other kinds of insects that we see on the land.
So I think the reason for absence of flowering plants and absence of insects are also
connected. So, but yes, well, that's another long topic.
I have a question relating to identification.
Yeah.
Either Subbu or Geetha can answer that. So this is the fact that now naturalists are armed with digital cameras, and that really has actually drawn them to various groups that were, so to say, inaccessible previously. So as with moths, if you were to identify them to species level, as Geetha has pointed out very clearly in the book, in the Blackbuck, it requires dissection and possibly microscopic examination, which is, I mean, that data is not available from a digital photograph. So, are there any efforts now over the last few years or several years since digital photography began towards what I think of as photo taxonomy, to steer the traditional identification characters to what is available from photographs? I can think of various reasons why I would not like to collect and preserve and so on. So that's where the question arises.
Would
Okay,
you go with
okay.
that? So, Mr.
You go
Subramanian,
ahead.
then I'll answer.
Yeah, no, you go ahead.
See, it is quite true. I mean, in the case of moths, even when you are able to analyze the morphological feature of his genitalia, you still find that you're unable to identify it, because sometimes in different species of moths, you still find anatomical features to be almost similar. They're very secretive. And so increasingly, scientists are resorting to molecular work. In fact, I think Mr. Subramanian would be able to tell you this, that today, for identification of species level, people, hardcore scientists are not even satisfied with morphological work, analyzing genitalia. They want to look at molecular work, you know, that at the level of molecule, because they are not merely identifying it through features, but they are identifying it through how evolutionally they have diversified. When did they come into being? When did the group originate? There's a lot more of work that taxonomists are doing, which is all right if you're a scientist and you're a researcher and you're very keen on finding out new species. But I feel that what we need today is habitat protection. And for habitat protection, you need to know what kind of insects are there. So personally, if you ask me, I don't much care whether an Autocaris physalis or an Autocaris amethystina, as long as I know there is an Autocaris over there. And then, okay, probably there may be one similar or different species. It would help me to say that, all right, this particular moth has some interaction with the plant or some other feature in the habitat, and therefore can we conserve the habitat? Today, rather than going into, of course, we do need taxonomy. We do need to know what species are. I'm not saying no to that. But I'm saying as a citizen scientist, somebody armed with a good camera and who's taking photographs, one can take photographs with as much details as is possible, because you need not collect a moth, but you can always pick up and keep your moth on a finger or on a little bit of stick, and then turn it 360 degrees all over, try and take a photograph of its proboscis, try and take a photograph of its front leg, the middle leg, the hind leg, look at it from a lateral view and see whether you can take the side view of an abdomen. You can, there are cameras are so good that you can actually collect a lot of details that might still help in identifying, at least providing a possible ID, a provisional ID for that. I know, because I don't collect insects. I haven't collected any insect or any moth so far for any of the work that I have done till today. And at one or two places, like for example, in my own house, I see moths, and nobody's going to stop me if I want to collect. But every time I have gone close to picking it up and putting it into the killing jar, something has stopped me and I have not been able to kill it. So I guess there might be other people like that. My entomologist friends tell me that that is the wrong thing to do, that you really need to collect them if you want to do some honest work. I call it honest because then you have to be honest with your taxonomy and what you're saying is the moth's name. But I think photographs can only give you limited information, however good you do it. You will need to collect it and you will need to do either some anatomical work or molecular work if you really want to go deep down and do good taxonomy. Have I answered your question, Kumaran?
Partly, I think, Geetha. I mean, my question also is related to what we should do about this in the future, because there's going to be a lot more digital photography coming and potential information being lost. I mean, I agree with you. Cameras are improving, and I dream of the day when they can do
They can look
three-dimensional
through it.
scanning of moths from on an ultraviolet screen, and that gives you maybe there can be X-ray scanning of moths with a handheld device.
But
I dream of that,
still,
but I'm just asking you what is happening right now.
what is happening right now is only molecular work. You want a little bit of its leg or a little bit of its tissue from some part of its body so that it can be done. Am I right, Mr. Subramanian? Dr. Subramanian? Correct?
Yes. So, Kumaran, I will tell you, there is the taxonomy works with a different philosophy. We are all human brain is a taxonomy, works as a taxonomy, because we name every object, whether it is animate or inanimate object, we name all the object and we make relationships, like consciously or unconsciously, we make relationships. So like, for example, we have thousands and thousands of models of vehicles on the road. And there are some people who can identify each and every model and each and every variants. Okay, so it's the same like a taxonomy. So they use same methods in taxonomy to identify all the variants, all the models. So human brain is I say, like, by construction, by design, is a taxonomy. So you have, like, when human beings evolved, so they had to classify edible and poisonous plants and animals, and they also have to identify enemies, predators. So that is how like our brain developed. And say, for example, we have, like, if you look at the names for our traditional names for our biodiversity, so most what is being used or what is being regularly interacted, so we have names for all those, so all those organisms. Say, for example, you hardly have a name for moths, common names, even either in Tamil or Malayalam. You like, recently there are attempts to make Malayalam names for moths. So they have, people who are successful in making several, but they are not in use. So they are now recently coined. But if you look at which are regularly hunted, or which are, like, for example, fishes. So you have the local names for all the fishes. So that is how our brain works. And like, we have group names, like, for example, moths, we have group name, or like insects, we have group name. So we don't, like, collection of species. But the taxonomy, like, over the years, say, like, it's a growing science. No? So species is a hypothesis. So species is a working hypothesis. So like, that is like, and the purpose of the taxonomy is to prove or disprove that species hypothesis. So each and as the technology grows, so people will identify more and more character and come up with more and more methods to either disprove or prove that hypothesis. So the taxonomic revisions are such studies which are establishing the species concepts, and the species concepts are also regularly changing. Now, like, it's a highly dynamic system, and now people are realizing that many species, what we like, including human beings, are hybrid species. So by this deep genetic analysis, like, human beings is thought to be a hybrid of Homo sapiens is thought to be a hybrid of 3 species of Homo. In the last 200,000 years, we have hybridized with other species of primates, other hominoids, and we have become as now what we call it as Homo sapiens. So this is also finding to be true for several species. Say, for example, like, Geetha has also illustrated in her book about the possibility of difficulty in identification because of polymorphism. So there are several moth species that is highly polymorphic within its geographical range. So they look different. They look as different species, but if you take photograph or illustrate, or even if you, unless you do a careful study of their characters, or you do hybridization experiments, or you do grow them, so you see that they are highly polymorphic. So photography is not going to be answer for all the organisms. So if, for example, maybe it will work for large groups, even for frogs, which are large vertebrates, or fishes, photography alone cannot solve the problem of species identification. So you have to go for analysis of characters. So now people are doing even micro CT scans. So micro CT scans are methods. You can, without dissecting, you can look at the anatomical features. So you can make a 3D construction of an anatomy, and you can study and you can know what is inside. So like micro CT scan, you can look at different systems. Like you can look at the muscular system, you can look at the nervous system, you can look at digestive system, and you can look at skeletal system, and you can even a small insects like an ant, you can do micro CT scan and you can study. So the technology, as the technology is growing, the species identification is going to be complex process, and people are not going to accept just by photography or by just by looking at few characters. Species, new species of proposals are not going to be accepted in future. So you need multiple lines of scientific evidence to prove a species. So that is going to be the future. So even a bird taxonomy is becoming very dynamic. So several subspecies of birds, what we are thinking as a geographic variants, will in future will be elevated to species. So it will be elevated to species because of all these molecular studies. So,
actually, a question that never gets discussed amongst taxonomists is, why are you spending so much of money on trying to prove that two similar looking moths or dragonflies are species? Can this money instead be used in the conservation of habitat? This is a question I've been asking lots of people. Maybe you could answer this, Dr. Subramanian.
Okay. So let us, I'll take the example of langur, the common langur. So earlier, so all entire India, it was supposed to be a single species of langur. I think it was called Seminopithecus centellus. Earlier it was called Presbytus centellus. So it was single species. So it was called a northern, there was a northern subspecies with a different tail carriage, and a southern species with another tail carriage. So, but molecular studies and even earlier detailed morphological studies put them into 8 species. So like in the Western Ghats itself, you have 3 species of langurs. So like you have this priam, you have these different species. So what happens is that, so now suddenly you have, when you have a single species of langur with a large geographic range in India and Sri Lanka and other neighboring countries, they are become like least concerned because they have such a large geographic range. So they are found everywhere. So people are not bothered. But when you do scientifically prove that no, they are not actually different, same species, they are different species with restricted habitat requirement and also restricted geographic range. So now you have their habitat. There is a much more arguing point for their conservation of their habitat. Now what happens is that suddenly all the dry habitats on the eastern side of Western Ghats, like where our priam, the Presbytus priam, especially the dry, you can see in Kalakkad, Mundandare also. Mundandare you can see.
Yeah, Tirunelveli side, yeah.
Tirunelveli side, or even in up to Mudumalai, Bandipur, you can see. So all those habitats like dry thorn forest habitat becomes a habitat for this because they are not found in wet forest. But in evergreen forest, you have another species of langur. So that is why like there is a like it's also kind of a scientific inquiry that okay, what actually is a species? So whether it is a different species or it is a, so the why they are trying to prove or disprove is that there is some variation. Either it may be a host plant, like for in the case of moths, it may be difference in their host plant associate. So one population of moths may be specializing in one host plant, and another species, another population may be specializing another host plant. So that is itself is an indication that, of course, there are some polyphagous species, but many monophagous species, there is an indication that, okay, this could be a new species. So let us investigate. So that is how it goes on. So there is a like, of course, that is the only way we can characterize the habitat also. Unless.
Yes, but you know, that seems to be in the case of moths, because in their, if you look at their numbers and their and the variations in them, there is still another argument, which I don't think we should go into here right now.
So, like for example, if you like now the environmental destruction is so much that we don't need even species identification to conserve habitat. So you can.
That's what I'm trying to tell you.
You
That's
just
precisely my point.
You just require a good satellite imagery, satellite studies to identify habitats and
the numbers. You need to know the numbers. You know,
numbers,
are there?
No, initial steps would be to conserve large areas of natural wilderness. Then you can go to, because this is an endless process. Okay, so this is endless.
That's a point. Point is that today habitats are disappearing at a speed that I don't know whether we will be able to catch up trying to do taxonomic studies. They don't match at all.
Yeah, that is true. That is true.
So therefore, if a particular patch, a habitat has got enough biodiversity, diversity of creatures, not just moths, butterflies, insects, nobody is looking at invertebrates. Nobody is, for example, looking at how many centipedes or millipedes are there. There's nobody studying millipedes, even so if there can be a diversity studies, and then, you go down to the species, I still think habitat should be the focus.
So for that also, like habitat characterization also, this molecular studies is helping. For example, there is a method called eDNA. It is called environmental DNA. So you can take from a pond water and you can estimate how many species are there.
Yes.
So like species identification, maybe like you may not have voucher samples or something, but if you have,
the diversity of DNA you're talking about.
No.
Through the species present over there.
Yeah. So you can take a sample of water and you can do this eDNA studies, and you can say like these are the species found, because for many species already DNA sequences are there. So you can match the sequences online and you can see. Then there will be unidentified species also. So you get from bacteria to mammals, all the species that is there in the water, you get environment, because all of them release some fecal matter or some dead body. So from all those things you can extract DNA, and these technologies are developed. So there is something called eDNA studies. They're expensive, but people are doing eDNA studies.
They're very expensive, isn't it? Molecular work is quite expensive.
No, expensive, but now they come down, drastically come down now. Because now you can get a sample done up to sequence level, only some 1,200 rupees, 1,300 rupees, you can do a sample done. So like there are several private companies who do it, very good things they do. So they up to like from, you just need to give them a tissue sample and they can give you the sequence, and even analysis they will do and give. So it is not just, the prices have come down very drastically. So in future it will come down, it is going to come down. These things are.
We're back at Kumaran's question on camera.
Yes.
Are there any moth parks around India?
Moth parks? No, I don't think so.
Okay.
It's largely because although there are several day-flying moths, a large variety of them are nocturnal ones. And the best way to see them is when you have lights on. They're attracted to lights. So I don't know. I've always suggested to the KMTR FD variously that Kudravati should be declared as a moth sanctuary, because that's the best place where you can see the most divergence of Western Ghats moths over there at any time of the year. But I don't think we have a moth sanctuary, leave alone a moth park. No sanctuary dedicated to moths. And it'd be good to have one dedicated to moths.
If you have a good, if you have a good plant diversity, natural diversity is bound to have a lot of moths. But
Kudravati is a place where there are lots of moths.
I know the Kudravati thing. I was discussing other day with Kumaran and Sudhagar. So the trick of moth watching is that you need to have a vantage point. So Kudravati is a vantage point. So you have the entire valley below. So what you have is that you get all the moths that is there in the valley. So they come up. So, and the best time to watch moths is during the new moon, days around new moon. So also that is the big thing we do around new moon. So there is no light. And there is a light called dark light. So many of
Yeah,
you
nowadays they do dark light mothing. Yes.
so that's it's a UV light. So that is how you get all the moths attracted to you. So if you put regular fluorescent lamp or mercury vapor lamp, now it's very difficult to get, but mercury vapor lamps are also very good to get. And if you have a portable genset, nothing like that. So that is how we use trapping moths. So UV light is the best. So like get a lot of moths in the UV light. So you need a vantage point. So we always go for hilltop, and when there is just light rain is very good. So if you have a light rain and hilltop,
Misty nights are the best.
Misty, and because
the geometrid moths come in large numbers on a misty night.
Yeah. So
they come after about, at Kudravati, they will come only after about 12 o'clock. There's a huge, that's when the mist moves in, along with that. They come in hordes.
No, they have flying time. So like they have they have very good rhythms. So the large moths, the bombycids and sphingids, they all come after 2 o'clock in the night. So if you start at like 6 after sunset, you can see like all these pyralids and all
smaller ones.
Small ones will come. Then Cambridge and all these things come. Then after midnight, you
the noctuids come in between. They are.
Yeah. And you get after midnight, you start getting all these big moths. But the good thing about moth is that once they settle down, they don't fly away. So in the daytime, they cannot see. The daytime, they are blind. So you can do it. And there are places in Kerala where we have recorded, like with Valapil, I have recorded in Wayanad, 700 species in a day. In one single night, we have recorded 700 species.
I have more than 1,000 species at Kudravati.
Yes.
One night.
One night.
More than 1,000.
Yeah. So one you can easily get in such places. So Kudravati you can get.
Kudravati, Upper Kodayar, Sengalteri, 3 places where you can get diversity and very beautifully colored moths.
Yes.
Even Upper Kodayar.
Upper Kodayar, yes.
Yes.
At Upper Kodayar, that EB guest house is the best place. So you can get a lot of moths. So there's a lot. I think they have this Malabar Natural History Society every year. They conduct this moth week. So in the monsoon, just before the heavy rain starts, before June 15th, they do every year, either in different places in the Western Ghats, they do this. So that has become a lot of interest in Kerala part. For studying moths, there is a lot of interest.
Very good. There's a question: are there any moths which are endemic to Tamil Nadu? Geetha?
There are endemic, but yes, if you see the list that Hampson himself has brought out from Nilgiris, there are several which are endemic to those regions.
Okay.
I have to get that list out because I can't tell you from the top of my head.
Okay, that's fine. That's fine. I'm sure there would be. And there's a question. Can you go to the chat box? This says, I often observe in my Chennai house in banana tree, coconut tree area, a small white fly size 3 to 4 mm appearing like moth when the photo is enlarged. What is the idea of this insect or moth? Is it Alirodicus rugio?
They are not moths. They are these aphids, no? That white flies.
Aphids, yeah, they are aphids.
Yeah.
When you see something white, they could be aphids or there could be some other species of insect. And a lot of, if your environment is very good, then you will have certain other insects that almost look like micro moths. Those are the caddises, the trichopterans that appear. And if it is a fairly good, some area with water nearby, you get lots of them which often be mistaken for small moths.
Yeah. Okay. So those are the only questions, and I think Sudhakar, would you like to say anything before we close the meeting, Sudhakar?
Hello. Can you hear me?
Yeah.
Oh, I wish to thank Dr. Subramanian and Dr. Geetha for wonderful morning, which has opened up my mind. Like I think everybody has got their vision widened considerably. I mean, I was enlightened by the whole discussion. Thank you very much, Dr. Subramanian. Thank you very much, Dr. Geetha.
Yeah.
And for all our members, have a good day.
Thank you all.
Yeah, and thank you again. The interaction that happened between Dr. Subramanian and Dr. Geetha. I mean, it was although it was an answer to Kumaran's questions, I think there were so many things that emerged out of that interaction, which was amazing, completely unexpected. Thank you, Dr. Subramanian. Thank you, Dr. Geetha, and thank you all members for taking time out and attending this monthly meeting. Hopefully, the next monthly meeting, because the schools have all opened, it is quite likely that I will be able to get permission to meet physically at our Alcott Memorial High School, Higher Secondary School. So let's hope that happens. So till then, take good care, be safe, all of you, and have a great Sunday. Thank you.
Thank you. Bye-bye.
Thank you so much.
Thank
Thank
you.
you, everybody.
Thank you.
Excellent
Thank
session.
you. Thank you, everyone.